Let's talk cephalopods. Everybody's favorite mollusk, right? Yeah, sure. Yeah. Here's Plectronoceros. This is the first confirmed cephalopod that has all the features we expect. We only have the hard parts, the soft parts, and the colors are reconstructed, and they are awesome. And this little video above has a little school of 3 Plectronoceros running around and doing stuff and just looking great. Absolutely great. Yeah, look at them. They get frightened by a priapulid worm, and then they kind of run away from an Anomalocaris later on. I think that's what that green thing is. All right, anyway, let's talk cephalopods. So these are the major groups of cephalopods. The ones you're probably most familiar with are the coleoids. And coleoids either have no internal shell or they have no shell at all, or they have an internal shell. Kind of like squid pen. I don't know if you've ever had a bird, but you can buy one of these things. It's the internal bone from a cuttlefish, and your bird can peck at it and be as happy as a bird can possibly be, I guess. There's also the chambered nautilus in this group, which is a fantastic choice if that's your favorite cephalopod. And then we got all the cool extinct ones. Now, cephalopods had a bad time at the end of the Cretaceous. See, there were a lot of groups that were doing really well, like really, really well, the ammonites, and they just get hammered at that point. So you got your ammonoids, including the ammonites, you got your ceratites, and you got your goniatites back here. Those are all like steps on the way to the same thing. And then they all just completely get crushed at the end Cretaceous extinction. We think these were top predators, so they do really well when there's abundant food, and when there was no food at all, like after the end Cretaceous, then they do not do well at all, and they just die in massive numbers. It's a live fast, die young kind of lifestyle, and surely you can respect that. You got some weirdos. That should be Bactritoids, Bactrilloids, yeah. Somebody needs to go to typing class. And your stem group cephalopods down at the bottom. There we go. So the thing we're going to find in the fossil record over and over is going to be mostly the shell, when there is a shell. And things that don't have a shell make very rare appearances in the fossil record. The living chamber up here is also known as the phragmocone, Phragmocone. Yeah, crazy word, but worth paying attention to. This is the part that's not supported by internal chambers. It's the outermost chamber where the organism was. It's rarely preserved because it tends to be just sort of the thinnest shell that was just recently made, and it's got nothing holding it up from the inside, so it gets crushed. And rare when you get a nice complete phragmocone on a fossil, but very nice when it does happen. Inside, you're going to have these little chambers. So it's going to divide itself into chambers, and every time it grows, it's actually going to make some more shell up here and then move up and then put a wall underneath, but leave a little doorway. And so this little path that connects all the previous living chambers is going to be the siphuncle tube, and there's an organism called the siphuncle that goes down that tube. And that is going to be instrumental in both pumping water into the shell and out of the shell, apparently. So, let's talk siphuncle, because it's like the defining feature of what separates cephalopods from other groups of mollusks. So it's worth a little time. Here is the one from an extant mollusk. So this is the chambered nautilus. And you can see its siphuncle here. It's got this awesome texture on the surface. Look at that surface area. Now, if you've done a little anatomy, that looks kind of like the surface of your gut. And the gut is maximizing surface area for absorption, and that's exactly what we're doing here. Because one of the siphuncle's jobs is to pull the water out of those chambers. And it's going to do that, we think, mostly by passive diffusion. There may be active processes that complement this as well. So the passive diffusion part, the idea would be that if you have very salty water inside of the siphuncle, then that would just draw the less salty seawater. Yeah, it's going to be saltier than seawater. From the chambers in, presumably leaving some of the salts behind. and then you could get rid of that. That is one idea for how this works. Inside of it, if you look at the cross section, there's a big vein running down the center, and in the nautilus there are 4 little arteries. You can tell the difference because an artery, look at that, it's got a big thick sort of wall around it compared to the vein, which has a much thinner wall. And so if we get fossils that are going to show something like a siphuncle, we can look for it. We know where to look. It's going to be in the siphuncle tube, and there's going to be a sausage-like thing in there connecting up all the chambers. And we want to look at the cross section and see what we can see. So, did we find some? I didn't, but Royal Mapes and Co. did. Here they are. So when you find a fossil of a cephalopod, it's really good to look for the placement of the siphuncle tube, more properly, and you can see it right there. And so if it's in the center of the chamber, then what you're holding is going to be something like a nautilus. If it is right at the edge of the chamber and you've got some degree of coiling usually, then what you've got there is something related to an ammonite, although there are exceptions. There are other groups that have a siphuncle right at the edge, some of the minor groups, and including these guys, bactritids or bactritoids. All right. The difference with those is that they're going to have an internal shell, whereas ammonites are going to have an external shell. All right, so here are fossils of a siphuncle in a siphuncle tube. So here's the siphuncle tube going right around there. This little SC thing is the siphuncle. Here's the surface of it. Looks a little different than the nautilus, but then this is an ammonite, not a nautilus, and this is a fossil. So this was wonderfully preserved by phosphatization, which is a way that you get some really good fossils. And presumably this would have to have been etched out with acid treatment to get it out of the rock, and it's gorgeous. But you can still see a lot of surface area there. It looks like a churro. Yeah, I guess a churro is maximizing surface area for flavor or something. I don't know. Here's another look at what looks like a different one. And if you look at the cross section, you can see what they've marked as a little vein and a couple little arteries up here. Here's a close-up on one of the arteries. So this is siphuncle-like. And we're in an ammonoid, so it may not be the arrangement of 4 arteries around one big vein in the center. But, you know, also these things may become less obvious after death has set in, because death always precedes fossilization as far as I'm aware. And you may get some degree of decay, which would make it harder to read some of the features inside. Here is another. This was published a couple of years after the previous study, and it's fantastic. This is another ammonite. You can see a beautiful siphuncle tube here. That's the ST thing. Look at that thing. It's even got little supports that hold it onto the surface of the shell. It's right near, this is the external surface of the shell, and so if it's right on the outside, that tells you this is going to be something like an ammonite. It's going to have this on the outside, and then that's the siphuncle. And look at that big central vein. It's collapsed a little bit more than it would have if it had blood pumping through it, but this hasn't had blood pumping through it in, I don't know, 100 million years, 150 million years, something like that. This one also has 4 little arteries in sort of a cross pattern around the outside. So, very similar to a modern nautilus. The surface is very lumpy-bumpy. It's not quite as easy to read, or it's not quite as maximizing of surface area as the nautilus, but that may be something that was lost on preservation. But yeah, another excellent, excellent fossil. These are once-in-a-lifetime things. Yeah. Okay. Cephalopods in general. Fantastic art by Franz Anthony. And look at these things. So on this row, it should be standing up tall, but it didn't fit. You can get it bigger this way. So we got some coleoids down here. We got some things related to ammonites and ammonoids in this row. We got some early branches up here, and we've got some nautiloids over here, number 3 and 11. Yeah, so let's look at these a little more. Okay. Here's our little friend, Plectronoceros, and I put a picture of one of the actual fossils, and this is a section through the fossils, so you can see the little chambers in there. That's these little things that are stacked up like that. And can you spot where the siphuncle is? Look, see all the chambers stop here, but the outside part of the shell is right there. So this little gap right there connecting up all the chambers, that is the siphuncle. And it's filled in with rock now, but at the time when it was alive, it definitely would not have been. Yeah, so that was a place where water could go in and out, which would allow the organism to go up and down in the water column. We've also got a couple of real weirdos. So here's Phragmoceros up here. This one is covered over in the middle. It's got a hole at one end where the head would come out, and it's got a hole at the other end where the siphon would come out. So it's going to look something like the little tiny, possibly monoplacophorian, these things called Eucalcionellids and Halcyonellids. They look like little turbo snails. I don't know if you remember those. But this one is a cephalopod. It's got chambers, it's got a siphuncle, and Sphoeroceros, shown down here, is a coleoid. Now, coleoids are going to have mantle, like think skin, maybe some muscles and other tissues in there, that's going to cover over their shell. So the shell's going to end up internal rather than external, like it is in the other two groups and in these guys. That is a thing like this. This is the group that today would have the sort of pigment cluster cells, and would be able to mimic, its background or use its behavior and coloring to mimic other organisms. No idea whether this particular one had developed that ability yet, but at some point along the way, it probably did, because it's pretty widespread in the existing members of the coleoids. So there you go. So here is that siphuncle from, it's a thing like Plectronoceros, sort of a different genus, but in the same part, the same group, I think the same family. And you can see there's our siphuncle, here's our hole that runs and connects the tubes, and it's got this piece. So the piece is shown in green here, and they've cut it away. This is what a complete ring would look like. And so every time they're laying down one of these little floors, one of these little chambers, they leave this hole in it, and there's this special piece of shell that forms a loop there. And that is going to be the loop that is the siphuncle tube through which the siphuncle is going to go. So it can empty out or potentially fill up those chambers. Here are two lovely nautiloids. Look at this. This is a little more like the modern chambered nautilus. This one is not at all like the modern chambered nautilus, but internally probably very similar. There are ideas. So I think the prevailing idea in the field right now is that these little spines wouldn't have done much in the way of dissuading a predator that was large enough to sort of chomp and crush the shell, like the ones that are around at this time would have a hard plate on the top of their mouths, I think, and so probably wouldn't be dissuaded as much by this. But there is some stability advantage, so it rocks less, and it's easier to sort of level out when it has little keels on it. There have been some nice functional studies that have examined that. And so it may be that that's what Cooperoceros was trying to do. And Cenoceros up there is just looking great, being its best nautiloid self. All right. There are ammonites in the world. These are what are called heteromorph ammonites. And so these are ammonites that are the exceptions. They have the weird shape. The ammonites you know are going to be over here like this. We'll come back to them. Let's just bask in the heteromorphs for a moment. So heteromorphs are going to have a shell that is not just a simple coil. It's going to have open spaces in it. It may change coiling at some point. And when it changes coiling, like when it's making the small shell, the head would be up. It would essentially be at this end. Then when it puts that little turn in, then all of a sudden the head is going to be here, and it's going to be looking at most of the rest of its shell. So it's basic, like the way that it works, its hunting orientation is going to have to change several times as it is growing this convoluted shell. This one looks like the shell is soft and it's just a twisted up ball of rope. It's not. It's hard, and it is just coiled that way. Doesn't seem like the most efficient way to build a body, but you get this sort of robust bowling ball of a shape, and I have no idea what the advantage of this would be, except that it worked well enough that we have a decent number of fossils of these. So, yeah, and those others, although they do tend to break. Yeah. So, ammonites you can always recognize because the siphuncle is right on the edge. It's going to be right near the outside edge of that thing. So on the center where the part, this is the keel, the bit that runs around the outside, that's where you would find the siphuncle, just underneath that. If you get ammonites and they are missing the outer shell, which frequently happens, you'll be able to tell because you'll be able to see all the little chambers in there, because you can't see those from the outside normally. And they usually don't match up well with the ornamentation on the outside of the shell. Yeah, very confusing creatures, inside versus outside. But if you get one that doesn't have the external shell on, so it's an internal mold, there will often be a groove that runs around the outside, and that'll tell you that you have got an ammonite in your hand, or something close to an ammonite, like an ammonoid. Yeah. Ammonites got big. This is one that's a normal sized lady there, and this is not as big as the ammonite would have been, because remember, the phragmacone, the living chamber, rarely fossilizes intact, and this one is at least partly missing. In a lot of ammonites, the phragmacone goes a little more than halfway around the shell, so even if you imagine it starting right here, it would still have gone around to about here, and so it would have been as high as the top of your screen. Ridiculous. Yeah, that's a lot of ammonite. And something that big that is a top predator would have needed to presumably eat a lot in order to just get the energy to, I don't know, swim around and be as awesome as it is. Yeah. Okay. Let's talk coleoids. I found a pretty picture of some coleoids over there, and so animalfact.com, hats off to you. Shown here, this is from a paper. This is looking at the points where each of these groups, the extant ones and the extinct ones, has split off, and so you have your nautiloid here just to root the tree, and then they're saying they're all coleoids from this point, which is going to be in the Devonian. There would be things leading up to this that would be coleoid-like, or may have been found after this paper was published. I'm not sure. Actually, no, definitely not found after this paper. So, yeah, and so these are going to have internal shells. The internal shells will sometimes have chambers, and yeah, the question is, are the chambers going to have sifuncles? You'll find out later on, but I'm not going to give that away right now. We got to keep you on the edge of your seat, right? So coleoids are not the only mollusks that have mantle that covers their shell. So one other one that's common in seashell shops is the gastropod called a cowrie. Shown here, and this is, I think, a leopard cowrie, I think they call it. But look at that. When it's alive, its mantle can stretch up and basically cover over the entire shell. So this is its mantle, its foot, essentially, that is doing that. Yeah. And it's thought that this allows for the shell to be repaired. So if there's any damage anywhere on the outside, it's able to fix it. Whereas for little guys like this, if they get damage to that shell, they don't have any living parts other than the siphuncle, which doesn't repair anything as far as we know, that are in the top part of the shell, the abandoned part of the shell, which is really important for their life because it's their depth control mechanism. And so injuries to the shell may have been fatal. Yeah. Live fast, die young, like a cephalopod. Yeah. So here is another one. This is an unusual coleoid. So it has an internal shell, but the shell is chambered. And if you look at the chambers, look at that little hole right there that connects up. You can see the little tube running up here. Yeah, that's a siphuncle. And this time it's all the way on the inside edge, which is weird. For an ammonite, it would be on the outside edge. For a nautilus, it would be roughly in the center, although it moves around when they get to enormous size a little bit. Yeah, and there you can see the squid itself, called a ram's horn squid. Very neat little thing. Here are some fossil coleoids. So this is a fairly early, but it's one of the earliest complete sort of soft tissue preservation cases with a coleoid. And so this big long shell, it's like a big long cone-shaped shell with the little layers in there. And I don't believe there is a siphuncle connecting those layers. I think they're just solid. It's just growing that way. But this would all be under some sort of skin, so you'd have to imagine some little sort of flappy squid-like fins on the side, and the tentacles are really tensed up here. They may have been a little able to stretch out a little longer, and there you can also see it's got a jaw in there, and so the jaws are related to the little doors that snails will pull shut, like land snails and sea snails. In order to sort of close off the soft parts from danger, called noperculum. Here's another example of beautiful soft tissue preservation on a coleoid. And on these Jurassic coleoids, you can see that their tentacles have these little hooks in them. Now, that's a thing that you find in vampire squid as well. These are not vampire squid, but it's a similar adaptation. Maybe homologous, I am not sure. The other interesting thing that you can see here is these little traces that come up under, I think that's UV light maybe. And those are in the right position. We believe those are nerve bundles. And so it may be something like the myelin sheath that is fossilizing here, or the fact that it's a big, thick nerve bundle that's helping it to fossilize. Because a lot of the other soft tissue you can see is kind of gone. Yeah, the eyes did not do well, although you can still see, bits of them. But they collapsed at some point on fossilization. Here's another one. You can see the position of the ink sac. Inking goes way back. There are gastropods that can ink. This is ancestral molluscan character for sure. And there's a little bit of the ink duct, and here you can see some preservation of nerves. So the body did not preserve well, but those nerves did pick up enough mineralization that they ended up getting preserved, which is kind of cool. And here's a much more complete one. Here you can see the ink sac would be right up in here. Here's a little duct. And you've got some organs. You got some preservation of at least one of the eyes is doing well. This is maybe the other eye that's sticking out over here. And you've got the start of the tentacles, but most of the meat of the tentacles has either been scavenged or rotted away. I think if it was scavenged, this would not be in as good a shape as it is. But there you can see the internal shell running right up the middle, and some of the soft tissue that goes around it. So there you go. That's been our little introduction to the different types of cephalopods, and then we'll go into the evolution of cephalopods in the next section. Come on back.